Hi everyone, this is Jonathan Cedillo, student at Grand Canyon University. And today I'm presenting to you the action potential in cardiac contractile cells and blebistatin inhibitor action. Presentation for the class Biology 520, Concepts of Human Physiology 2, Dr. Joan Tassoni. Blebistatin. With the Scholar article, the effective use of blebistatin to study the action potential of cardiac pacemaker cells of zebrafish, Daniel Rerio, during incremental warming, the molecule blebistatin is studied. This molecule is a potential inhibitor of actin and myosin molecules, which blocks muscle contraction of cardiomyocytes, even after action potentials have been initiated. With the animal model zebrafish, data collection of the activity in pacemaker cells located at the sinoatrial region of the model's heart allowed investigation of the mechanisms of blebistatin use to modify pacemaker cell electrophysiology. This investigation was carried on using different parameters of action potentials after analyzing them. However, action potentials did not have a noticeable change even after the addition of the inhibitor. In contrast to a control group, blebistatin treatment group showed small changes in the activity of diastolic depolarization and repolarization. This is only suggesting that the inhibitor molecule of interest allows excitation contraction uncoupling that does not modify axial potentials initiated by the pacemaker cells located at the sinoatrial region, which is used in zebrafish for studies. Background. The zebrafish is a reliable model to study the mechanisms of the physiology in action potentials in cardiac contractile cells. Cardiac action potentials in zebrafish are initiated at the sinoatrial region of their heart. Besides the use of the blebistatin, other methods have been used to study the activity of pacemaker action potentials, but the techniques were given modified results. However, the inhibitor of interest has demonstrated that it can stop the activity of myocytes, which inactivates actin attachment. This evidence allows the authors of the articles to suggest that this agent is an excitation contraction uncoupler of pacemaker and electrophysiological activity. This only means that blebistatin uncouples cardiac excitation contraction that does not make significant changes in the actual potentials, which can be of trust when studying electrophysiological mechanisms in zebrafish pacemaker cells. Results of experimentation. The addition of blebistatin inhibitor into the circulatory system halted cardiac activity. By doing this, the agent applied was able to predict and analyze stable action potentials. In room temperature, the inhibitor did not show large modification on the actual potential firing rate of pacemaker cells. Depolarization, repolarization, voltage, time, and amplitude of the actual potential. Additionally, there was no difference in the threshold potential, overshoot potential, and maximum hyperpolarization, which might be indicatives of the changes that wave actual potential might go through. Furthermore, the constant rise in temperature upregulated heart rate activity together with the rate-dependent actual potential variables, such as spontaneous depolarization, actual potential depolarization, and repolarization. Inhibitor blebistatin did not show negative effect over most of the actual potential variables in all temperatures used. However, changes in spontaneous diastolic depolarization and repolarization at temperatures 26 through 29 degrees Celsius only were recorded. After computing all temperatures, scientists of the paper analyzed the data using linear regression, which, which showed that there is no important difference between the application before and after of blebistatin of the action potentials. In addition, confidence intervals of the analysis demonstrated that the variables of spontaneous action potential depolarization, action potential repolarization, and depolarization overlaid. The different temperatures did not show changes in the voltage-dependent variables between the control group and blebistatin treatment group. However, overshoot potential had a significant change in the inhibitor treatment group only at temperatures 26 through 29 degrees Celsius. Further, linear regression analysis of the temperature revealed that there were no compelling changes across the control and blebistatin treated pacemaker cells for each of the variables. Results. Here in figure 1, author of the article represent the variables in the pacemaker cell action potential. Number 1 represents the spontaneous depolarization time in milliseconds. Number 2 is the spontaneous depolarization potential in millivolts. Number 3 is the overshoot potential in millivolts. Number 4 represents the heartbeat period duration in milliseconds. Number 5 is the amplitude of the actual potential in millivolts. Number 6 is the depolarization potential in millivolts, which was recorded from the point of threshold to depolarization peak. Number 7 is the threshold potential in millivolts. Numbers 8, 9, and 10 represent the total repolarization potential. Number 10 is also the representation of the difference between the actual potential peak and maximum hyperpolarization potential in millivolts. Half of the actual potential repolarization potential of number 8 was recorded as 50% of the maximum repolarization potential in millivolts. In addition, half of the actual potential time frame was acquired from the actual potential repolarization potential and the point of depolarization of the actual potential. Here, table 1 shows a contrast between the actual potential variables of the control group and inhibitor blebistatin treatment at different temperatures, from lowest to highest temperature. These comparisons between the two groups were analyzed and statistically summarized using an unpaired two-tailed t-test. Compelling differences between temperatures, including room temperature, were recorded using one-way ANOVA. Comparisons across pairs of means were identified using post-hoc Bonferroni multiple means comparison test, on which the p-values 0.05 was considered significant. The number of hearts for each group and temperature is indicated by N. Furthermore, this table is demonstrating that at room temperature, the inhibitor blebistatin did not have compelling activity onto the actual potential firing rates, as the heart rate of pacemaker cells, including the rates of spontaneous depolarization, rate of depolarization, rate of repolarization, voltage, and times of diastolic duration, depolarization, and repolarization. In addition, contrast between voltage variables were not observed. This is only suggesting that there are not significant modifications in the actual potential shape. Additionally, the temperature did not make modifications to the voltage-dependent variables among control and treatment groups. However, the overshoot potential was importantly different in the treatment group at temperatures 23 to 29 degrees Celsius. Figure 2 shows the activity of blebistatin and temperature over the rate of diastolic depolarization represented by graphs A and B. The rate of depolarization is presented by graphs C and D. Finally, the rate of repolarization is presented by graphs E and F. Contrasts between actual potential variables at different temperatures are shown in graphs A, C, and E. Additionally, a linear regression analysis about temperature ranges is shown in graphs B, D, and F. Within the information presented, the points represent individuals. The means have been recorded together with their corresponding standard deviation. Statistics of the summaries between groups was recorded using a t-test on which p-value was significant at 0.05. Furthermore, the rise in temperature increased heart rate activity together with rate-dependent actual potential variables such as spontaneous depolarization, actual potential depolarization, and actual potential repolarization. The treatment group was not affected by the activity of blebistatin on actual potential variables. However, it did show small differences in the rate of spontaneous diastolic depolarization and repolarization across the 26 to 29 degrees Celsius temperatures only. The linear regression analysis suggested no compelling changes in the actual potential variables before and after blebistatin treatment. Finally, confidence intervals showed significant similarities when they overlapped at the rates of spontaneous repolarization and repolarization actual potentials between the control and treatment group. Figure 3 shows linear regression data about the activity of blebistatin and temperature over the rate of diastolic depolarization, shown in graph A, the rate of depolarization by graph B, and the rate of repolarization in graph C. The information presented, individuals in which solid color points represented recorded measures and open data points represented predicted values. After information was statistically analyzed, results revealed that the temperature did not have a compelling effect in the pacemaker cells of the control and treatment groups. In conclusion, the activity of blebistatin in the zebrafish heart positively affected uncoupled excitation contraction event. However, it did not modify AP variables presented in temperatures between 20 to 33 degrees Celsius. This is only suggesting that blebistatin inhibitor is a potential drug agent important in cardiac research regarding parameters of actual potential measurements. Finally, as an overall, blebistatin inhibits cardiac contraction without significantly modifying the sinoatrial pacemaker actual potential in the zebrafish. Thank you for watching. See you next time.